Um, So I think probably a lot of you are familiar with this already, but so I'll go through relatively quickly. The reason we care about natural products is because they are really important in drug discovery. So this review basically goes over all the natural products from all the FDA approved drugs from 1981 to 2019 and looked at where those drugs came from. And so about a quarter of them are unaltered natural products, botanical natural product mixtures, or natural product derivatives, which would just be a small chemical modification to a natural product. And then another quarter are synthetic molecules, but they were somehow inspired by a natural product, either through mimicking a natural product pharmacophore, or maybe the protein target was discovered through study of a natural product. So together, about half of drugs have some sort of relationship to natural products in their development. So that is kind of why we're interested in discovering more of these and making natural product drug discovery more efficient. And also, this is just an example of some different classes of natural products that we work on and what their potential applications are. So antibiotics, anti-cancer, immunosuppressant are all really common activities found in natural products. And these also really have really interesting chemical structures. So they also inspire a lot of chemistry, and also the discovery of the enzymes involved in this biosynthesis is also useful for biocatalysis applications. Um, so what makes a lot of our work possible is that in bacteria, the genes that produce these natural products are clustered together in what's called a biosynthetic gene cluster. And so that makes them relatively easy to identify. And we use a previously developed program that's really common in the field called anti-SMASH. And it works by identifying kind of some core signature genes for a specific natural product class, and then looking around in the genomic neighborhood for other potential biosynthetic genes, and then kind of looking at set distance out from the last one of those to set a boundary. And so this is just an example from this one genome, Streptomyces roseosporus. And so every yellow arrow here is a predicted biosynthetic gene cluster. Most of these are uncharacterized, but there's one here that makes an antibiotic called daptomycin that's been relatively well characterized. And so there's like core non-ribosomal peptide synthetase genes that are responsible for making this kind of core peptide. And then a fatty acid ligase gene that's involved in adding this fatty acid tail on, and then several other genes that provide additional support, like making some of these unusual amino acid precursors. They could be involved with resistance, regulation, and transport as well. And so since we can use anti-SMASH to find these gene clusters relatively easily, that kind of makes a lot of our future analysis on gene clusters possible. So one of the problems we want to address is that natural product drug discovery is really low throughput. And the throughput kind of narrows as you get further along in the pipeline. So it's really easy to run anti-SMASH on many bacterial genomes. You can do that for millions of genomes in a relatively short period of time. But then once you get to the actual structural elucidation and purification of a natural product, that is going to be really low throughput, and you're kind of working with one compound at a time. So what we want to do is develop computational techniques to make all these stages more efficient. Another huge problem is rediscovery, which I've kind of indicated by this dereplication step over here, where you're rediscovering known compounds. So if you have a way to prioritize initially for gene clusters that are going to make novel and bioactive compounds, then you can make the whole rest of this pipeline potentially have a higher success rate. And there's already some methods for comparing gene cluster similarity. So what we wanted to focus on was how can we prioritize which gene clusters are going to make a compound with a bioactivity that's useful for a therapeutic application. And so that's what I'll be talking about today. We also have some projects on some of these other areas, but I'll just focus on this one for time. And just to kind of further highlight this problem, there's way too many biosynthetic gene clusters for us to study. So this one paper clustered biosynthetic gene clusters into families that likely produce the same or related compounds. And then they kind of count how many of those there are. And these, again, are all just bacterial from bacterial genomes. So it's not including like fungal or plant or animal natural products. So there's even more than what's shown in this figure. And so the solid lines are the gene clusters they counted from sequenced bacteria, and then there's a projection with the dotted line for what would happen if you continue to sequence more bacteria, as well as some like metagenomic samples if you started to include more of those. And this top curve that includes those metagenomic samples, the authors of this paper estimated that there would be about 90,000, over 90,000 biosynthetic gene cluster families across all bacteria. And so that's way too much for a single lab to, or even many labs to study. So we need a way to prioritize what gene clusters we're interested in looking at. And so in thinking about how to make that prioritization, we thought a little bit about how people have kind of historically studied biosynthetic gene clusters and natural product bioactivity. And usually people think about how does the gene cluster determine the structure of the natural product, and then how does the structure of the natural product determine its function. But since these two arrows are kind of connected, we wanted to ask, can we predict natural product function directly from the biosynthetic gene cluster? You could try to predict structure first and then function, and we're interested in trying that as well, but that just kind of creates more opportunities for error because you could have error in predicting the structure and then additional error in predicting the function. Additionally, predicting the structure is going to be a much harder problem computationally because it's not a yes/no answer. Whereas the function, you can make that a binary classification problem where the molecule either has this activity or it doesn't. So it's just much easier to set up. There have been some efforts, for example, the program PRISM that tries to predict structure, and it works in some cases and not others, but our approach was to just try to predict the function directly from the gene cluster. So to do that, we built a data set of natural products with known gene clusters and known activity. This was based off the MyBig database. So that one has the gene cluster linked to the product, and then we had to do literature mining in order to find the bioactivity. And in total, that left us with about 1,000 clusters. So it's a relatively small data set for machine learning application, which is one of the challenges in this project. So then we make a numerical representation of the gene cluster, which I'll explain on the next slide. Then we train a classification algorithm for a specific activity of interest to predict, does it have that activity or not? Then we can use these models to make predictions for new gene clusters to prioritize which ones we want to study, and also identify genes that are associated with activity. So to choose these features, which are the basically elements that the machine learning model is trained on, we use the counts of different gene annotations, and we use anti-smash to find those. So those gene annotations are things like protein family domains and other similar domain classifications that give us information about conserved domains in the protein and what their function might be. And so I'm just using the erythromycin gene cluster as an example. So the genes are down here in green, and then the protein family domains and other domain annotations are up here in blue. And just to see what some of those tell you about the molecule, for example, erythromycin is a type 1 polyketide. And so it has different modules that are responsible for adding onto this carbon-carbon backbone of the erythromycin. So one of the domains that's important for that is the acyltransferase domain. So by counting those, we can get a sense of how large a polyketide, type 1 polyketide would be. Then there's also a ketoreductase domain. That one reduces this position here. So by counting those, we can get a sense of how reduced the molecule is. We can also count things like glycotransferases, which will tell us how many sugars potentially get added onto the molecule. And so we can get some very basic information about the structure that then gets fed into the machine learning model. And we hope that that is enough information in order to get some correlation to the activity and be able to make those predictions. The other thing we include are resistance genes. And so we use a program called the resistance gene identifier for this, for identifying these. And so the reason we decided to include these is one of the activities we're trying to predict is antibacterial activity. And so if a bacterium is producing an antibacterial, it needs genes to protect itself from that. And usually those are contained in the gene cluster. So for example, erythromycin has these two that were picked up by the resistance gene identifier. So we also count those and include those in the features. So here are the predictions. And so We use three different types of machine learning models for vector machine classifier, logistic regression, and random forest. And I'm not going to get into specifically how those work, but if anyone has questions about it, I'm happy to answer them. And in these graphs, the solid bars are real predictions, and the ones with the diagonal lines through them are negative control simulating random guessing. So we scramble all the features and train the model on just those randomized features. And that is going to simulate random guessing. And then we just look to see, we use that to see if there's statistically significant improvement over that guessing. And that's the case for all of our classifiers except for the logistic regression classifier for the antifungal case. And in general, though, it doesn't really make a huge difference which type of model is used. They all perform about the same within each classification task. So the classification tasks we have are general antibacterial activity, activity against gram-positive bacteria, activity against gram-negative bacteria, general anti-eukaryotic activity, activity against fungus, and activity against tumor cells or other like human cell lines. And our most accurate one is the antibacterial one. Our least accurate ones are the antifungal and anti-gram-negative ones. Those two are likely less accurate because we don't have as many examples in our training set of compounds with those activities. So far, our main focus is on validating that this antibacterial classifier can be used in practice to guide the discovery of antibacterial compounds. And then we're also at some point planning to expand our training set to hopefully improve the accuracy of the classifiers that are not performing as well. So we can also look a little bit just from the machine learning models to learn about how they're making their predictions and what kind of features they're using. And that can also potentially tell us things about what features maybe contribute towards activity. And these do differ by the different classifications, which is also encouraging. So just one example, so these pie graphs are kind of the broad classifications of these different genes. And then the top 50 are the ones ranked by the logistic regression model in this case as being most associated with the activity, and the bottom 50 are those least associated with the activity or associated with being inactive. And so for the antibacterial one, we have a lot of genes involved in ribosomal peptide and ribosomally synthesized post-translationally modified peptide synthesis, whereas those are missing from the top 50 in the antifungal. And this is likely because they're in our data set, most of the RIPs that we have are antibacterial and very few of them are antifungal. So you can get kind of these broad correlations. Another thing that was interesting was the transporters, which are this kind of like medium purple slice here, are really highly represented in the top 50 of the antibacterial features and less represented in the top 50 of the antifungal features. And this could be because since we're dealing with only bacterial gene clusters in this case, a transporter is a mechanism of resistance. So you need the transporter, the bacteria needs the transporter both to get the molecule out to its target, as well as to basically get resistance. And this is a very general mechanism of resistance. Some of these transporters are not particularly selective to specific structures, so they can kind of evolve to work for the product of the gene cluster more easily. And so that might be why these are the most represented resistance genes that are the most useful for prediction. And then for the antifungal case, you maybe still need a transporter to get it out to its target, but it's less crucial since you don't need it for resistance. So that could be why it's a smaller piece of the top 50 in that case. We can also visualize these predictions. And I apologize for the color scheme here. I'm still working on switching it to not red green. But this is just an example where you can have each domain. You can look at how each domain contributes to the ultimate prediction, and then sum over each domain to figure out what the overall contribution of each gene is. And so that's this shown for this one example. This gene cluster is predicted to make a glycopeptide antibiotic, which is like somewhat similar to vancomycin. So we expect it to make an antibiotic. We haven't verified this one in particular, but it is predicted to have an 89% chance of antibacterial activity. So that kind of lines up with what a human would expect from looking at this gene cluster as well. Some of the surprising things are that these long genes here are the non-ribosomal peptide synthetase genes. So this makes a core that is found in a lot of these glycopeptide antibiotics. So we would kind of expect the model to think that these genes should be associated with activity, but they're some of the most read or at least associated with antibacterial activity in the gene cluster. So that is kind of unexpected. And potentially the reason for this is our model doesn't have any way to know what order the domains are in. So there's no way for it to really figure out what the core non-ribosomal peptide structure is going to be. So that's something we're working on trying to make a new model that has that order information about the domains. So kind of by doing this analysis, we can pick out things that we could potentially improve. One other thing I want to highlight is that this gene highlighted here is one of the most strongly correlated with activity from the gene cluster. And this one is an ABC transporter, so that just further supports that the transporters are some of the most predictive features for activity, and one of the things the model relies on the most. So to get a better sense of how this would work in practice, we did a holdout set analysis. So this is where you have data that you have not included in the training set. And that is a better way to see how the model might perform in the real world. So also to further get information, we broke down that holdout set by how similar each gene cluster was to gene clusters in the training set. And we used known cluster blast score, which is a part of anti-SMASH to do that. So in these graphs, the ND are gene clusters that are not even recognized as gene clusters by anti-SMASH. So these are probably pretty different from the training set, and they're not one of the kind of like canonical natural product classes. Then we have increasing similarity to the training, to at least one gene cluster in the training set here. And you can see, as expected, the accuracy improves. And the cutoff is around 50 in all the classifiers, is around 50%, where you start getting improvement over guessing. So that's just kind of a guide to users on when to start trusting these predictions. For the antibacterial one, it is a bit better, and some of the less similar clusters still do okay. And just to give some examples of what a 50% known cluster blast score looks like, since there could be some concern that these would be very similar compounds. These two compounds have a 57% known cluster blast score between each other, but you can see that the structures look very different. And in this case, the model is correctly able to predict the activity of this holdout set molecule on the left, despite only having the most similar example in the training set be this one, this one on the left over here, despite the most similar one being this one, which is quite different. And in some cases where the compounds are similar, so in this case, when I built the training set, I only used the literature. I didn't use patents, only published papers or preprints. And so this polyoxypeptin was not described in the literature for antibacterial activity, but it is described in patents as being antibacterial. But I had it marked as not antibacterial in the training set. And this very similar compound, aurantomycin A, was in our holdout set data correctly assigned as antibacterial because there's literature describing it as antibacterial. And our model was still able to correctly predict that this is antibacterial, even though it had incorrect information for the similar example. So that further supports that the model is able to generalize to some level and pick out things, even if there's some incorrect information in the training set. So we also want to use this in practice. So to do this, we prioritize which bacteria we want to study by running predictions on bacterial genomes. So the bottom graph over here shows those predictions for each biosynthetic gene cluster in this particular Mycolotopsis aureus strain. And then the color of the bar just represents the predicted natural product class. And the higher the bar is, the more likely it is to produce an antibacterial compound. And then the top graph is that known cluster blast score I mentioned, which we can just use as a guide to get a sense of how many known compounds it might make, as well as how much to trust the predictions. So we got this strain, we grew it, and we do organic extractions on it. And all this work was done by a prior undergrad in the lab. And she was able to figure out, using bioactivity guided fractionation, what peak in our chromatogram was the active compound. So that was this one here. And so down here is the zone of inhibition assay. So the fraction collected here corresponds to this part of the plate, and you can see that it's inhibiting the growth of the bacteria relative to a solvent control. Here's the LC-MS and mass spectrum for this compound. So it's relatively small for natural products. That made the structural assignment a bit easier. And so at this point, a grad student in my lab finished up the structural assignment because the undergrad, Israel, had graduated. And so this compound over here is the more active one. And we also realized there's this other compound that's related, which is slightly less active, which is also produced. Unfortunately, both of these compounds are known already. So we rediscovered this known compound, but the gene cluster for these were not known. So there was no way we could have known upfront that this strain produced these known compounds. And so through this work, we're able to assign the gene cluster as well. So we're still providing some information, and this gene cluster, obviously, since it wasn't known, is not in our training set, so we can still use this to test how it works. So to figure out what the gene cluster is, we used these two compounds, which do have known biosynthetic gene clusters. So those are shown on the top and bottom here. And then we searched our genome for similar gene clusters, and so that's shown in the middle over here, where all the arrows that are the same color are genes with some level of homology. So then the question was the prediction correct? So this is the gene cluster. This is the prediction for that gene cluster shown here. So it's above the 0.5 threshold we use to assign whether something is active or predicted to be active or predicted to be inactive. So this was a correct prediction in that case. We're also able to look a little bit at how this gene cluster differs from the other related ones and find some potential new enzymes. I'm not going to go into this too much for the sake of time. So then just one final thing is, sorry, it's not, okay, there we go. Does this work for non-bacteria? So the first thing we've tested this with is fungi, and this work was all done by Olivia Riedling, who was a rotation student in my lab. So she did a ton of work during her rotation, and then she ended up joining a different lab at Vanderbilt, the Rokas lab, that works on fungal secondary metabolism. But we continued this as a collaboration and published it. So that was great to have this project come out of a rotation. So she found only 314 labeled fungal biosynthetic gene clusters. There's probably more out there, but this is what was easily kind of like accessible through MyBig and the literature. And so this is a lot smaller than the bacterial training set. And here is the results. We didn't do as many different activity classes for this one. And the blue is the real data, and the gray is the same negative control scrambled data strategy we used for the bacterial one. And so you can see that the improvements, especially for the antibacterial one, are not as great over random guessing compared to the case with the bacterial gene clusters. Weirdly, it kind of does a bit better with the antifungal activity, and then slightly better as well with the fungal classifier does with antibacterial. But still, this is not as good as the bacterial classifier. So then the question is, can we combine this data? If there are some similar features that determine activity between fungal gene clusters and bacterial gene clusters, then combining them should potentially improve the predictions. So that's what's shown here. And so the blue bars, the combined predictions, the orange one is bacterial alone, and the pink is fungal alone, and then the gray is the randomized one. And so for the antibacterial case, it does help a lot relative to the antifungal alone to include bacterial gene clusters in the training. It does hurt very slightly the accuracy of the bacterial predictions, but this is not really a significant difference. And same thing for the anti-tumor classification. And then the antifungal classification, which is the one we saw do actually okay for the fungal biosynthetic gene clusters, it doesn't really help or hurt to add in the bacterial gene clusters to the training set. So can this be used for plants? I think maybe, but it's going to be a lot more difficult and even more difficult than the fungal natural products. And so the main reason is that there's not as many plant genomes out there just because they're larger and more difficult to sequence. And then the biosynthetic pathways are not always clustered. They can be scattered through the genome. And since we need to identify in advance what what all the genes are in order to make the prediction, if you don't know the product and the genes are not clustered, then there's not really currently any way to do that. There is a version of anti-smash for plants called Planti-Smash, but That one is older and not as developed as anti-smash, where they're basically coming up with new versions of anti-smash every year. The planti-smash hasn't really been updated, so that might not work as well for finding gene clusters. But in general, I think the fact that combining the fungal gene clusters with the bacterial gene clusters improved predictions means that there's some hope for eventually someday developing this for plants. Um, and so just to summarize everything, we can use this strategy to help identify gene clusters that are likely to produce active compounds. If we had more accurate models, which I think we can accomplish through more training data, as well as some of the newer like pre-training model transformer strategies, we could make this more accurate and more efficient. And then one challenge we face in the validation is that if we want to test our prediction for a specific gene cluster, obtaining the product of that gene cluster is really difficult. And so one other thing we're working on in my lab is trying to predict what small molecules we can add to induce expression of a specific biosynthetic gene cluster. And yeah, I'd just like to acknowledge my lab. The highlighted students, Christine and Isra, were the ones who did the dipyramycin work. And then the collaborators I mentioned on the fungal biosynthetic gene cluster prediction and my funding. And yeah, thank you all for listening and for the invitation. I'm happy to take questions or discuss anything.
Fantastic. Thank you. Thank you, Dr. Walker. Any questions? You can feel free to raise your hand or simply unmute yourself and speak, or put your question in the chat and I'll read them aloud. Eric.
Yeah, great talk, Allison. Um, so clearly in plants, I think that the number of true gene clusters is significantly lower even than what has been anticipated. So I'm wondering to what extent co-expression could be a good proxy for having them in close physical proximity on the genome.
Yeah, so I mean, if you looked at co-expression and knew these genes are all being co-expressed and potentially part of the same pathway, even if they're not next to each other, then we can still like develop our predictions to, you would just basically take all the features from all of those genes and combine them into a count vector, even if the genes are not clustered. So yeah, some sort of strategy like that could definitely work to adapt this technique for plant biosynthetic pathways that are not in gene
Did
clusters.
you, yeah, did you do some, because you have the data from microbes to see to what extent co-expression could actually be a good proxy, since you know when they're in clusters and when they're not in clusters, you can probably get some ideas to whether that's going to work or not as efficiently.
Yeah, so yeah, we've done a little bit of looking at that for our elicitor prediction project. So we have seen there's definitely some, we've only looked at bacteria so far. There's definitely some correlation within at least operons on the gene cluster, but sometimes gene clusters are broken into multiple. And in those cases, like the correlation between different operons within the same gene cluster is not necessarily that high, but we haven't like formally looked at it across many gene clusters. So it's definitely possible that there is some correlation for the over the whole cluster. We'd have to look more at that.
Thank you.
I guess I have a related question. I'm sorry to jump in, but in that context, have you noted that some genes within a cluster are essentially like distractors or are like counterproductive to the accuracy of the predictions? And if yes, in like, are those some of the ones that are not co-expressed, or could that signal be, that signal being provided to the prediction algorithm be reduced, like the conflationary signal be reduced by incorporating expression data or some similar like orthogonal piece of information?
Yeah, that could definitely be the case. So, in our training set, like everything is coming from MyBig, so there's already been some work to figure out like what genes are really in the cluster. Whereas when we make our predictions, we just have the anti-smash predicted region. So there's definitely genes that are not actually in the cluster that get included as features in the prediction. So, Yeah, I think that could definitely hurt the prediction because we don't know what direction those non-cluster genes will force the prediction. So yeah, definitely using co-expression data to figure out like what the boundaries of the cluster are could help a lot. Yeah.
Other questions for Dr. Walker? Gaurav.
Hi, thanks for the nice talk. So I'm not sure how to frame this question, but I was wondering about the training data and basically in terms of classification as antibacterial or not. I mean, it's many times dependent on the dose of the compound. So if something is classified as not antibacterial, how do you apply a threshold for saying it's antibacterial or not, and whether the definition of the training data could affect that? And the second related question is there any signal in the biosynthetic gene cluster or the expression data to predict the dose at which a compound would be relevant?
Yeah, so that's definitely a huge challenge. So a lot of the papers do not report quantitative data on how active the compound is, especially older papers. So it's not easy to get like a data set that's all has that information about the what activity threshold you could set to define something as active or inactive. So as a result, when I was building the training set, I just kind of went with whatever the authors claimed. So if the authors said it was active, I counted it as active. But sometimes people kind of stretch the definition of active and the concentration they're using is pretty high. So that's definitely a limitation. But since there was a lot of times quantitative data missing or people using different assays, that's why I decided kind of not to initially go with any sort of threshold or quantitative output. But yeah, I think as we move forward and maybe get more data from more current papers that have more detailed quantitative data on the activity, then it would definitely be good to add, different predict at different thresholds, have different models trained for different thresholds, or maybe one that outputs, predictions at multiple thresholds, and then also have something that's regression instead of a classifier to predict a certain what the expected activity level would be. But yeah, I think the quality of the data is just like not quite good enough for doing that yet.
Yeah. I had one more question if there aren't any more. I don't see any in the chat. Um, have you thought of using evolutionary conservation of a biosynthetic gene cluster as a signal for identifying more important versus more important antibacterials or more important biosynthetic gene clusters overall?
Yeah, definitely we could look at how conserved the cluster is. And so I have seen that it varies a lot. Some gene clusters seem to be like very conserved across like an entire genus, whereas others are very rare. A lot of the most common ones have already been discovered, since it's easier to find them. But yeah, I think if we were to like do additional prioritization, then prioritizing for something that's very conserved but hasn't been characterized before could definitely be another way to try to get more active compounds out.
Other questions for Dr. Walker? I've got a whole list. Can I ask another question about the training data? Can you talk a bit about, so I guess did you generate it manually by inspecting abstracts essentially, or?
Yeah, so yeah, I basically read through like every paper and checked. I mean, does have links to papers, so that made it easier. In some cases, though, the paper they linked is one describing the biosynthesis, and I had to look to find a different paper that described the activity. So yeah, I first looked at the MyBig paper, then did like a literature search if the MyBig linked paper didn't have the activity. So yeah, that was very tedious. So another thing we're working on, we have a preprint on it, is using ChatGPT to more quickly scan these papers. And we're still doing it in a semi-automated fashion where we like give ChatGPT the PDF and ask it questions. But that's still a lot faster than reading it manually, I think. And then you can sometimes it does pretty well actually. But even once you have the output, you can even more quickly see like was that text actually in the paper? to just double check that the ChatGPT is correct. And it's definitely faster than having to scan through the entire paper yourself to try to see if it describes the activity of the product.
We've talked about a couple different things as far as improving your predictions. And one of the ones that you mentioned, of course, was expanding the training data. And something that's come up in the question session has been trying to identify signals of different types to improve the prediction, whether it's expression or evolutionary conservation. Between trying to increase your signal to noise ratio, essentially using different techniques versus expanding the size of the data set using like a language model to filter through a bunch of papers or something like this. Do you have a sense of which of those two, if it is one more than the other, is going to be more important in improving your predictive pipeline?
Yeah, I think it's hard to say without testing it.
Yeah,
That's a good question. Yeah, I don't really know if I can predict which one would be
that's
better. So
that's fine. I
yeah,
just
but we'll have to see and do some comparison. And yeah, we're working on now a little bit like how much does it improve when you start adding things. And it does seem like you really need to add a lot to get the improvement. So yeah, maybe some additional features like conservation might help more than than adding more, unless you could add like a lot more. Like I think we might need to double the size of the training set before we start seeing really significant improvements.
Are there any last questions for Dr. Walker? Well, thank you again for a great presentation and a great discussion. Thanks everyone for being here. Don't forget, we've got one more session in a couple of weeks, and we'll see you then. Have a great weekend, everyone.
Thank you.